Welcome to all our attendees. I hope everybody can hear me, and if you can't, please let us know. My name is Kavita Aswani. I am the Senior Application Scientist for Life Sciences at Lumen Dynamics. We're very excited to have so many of you join us today to learn the difference between the different light technologies for your applications. There are a lot of light technologies out there, and we understand that it's difficult to figure out which one you need for a simple yes or no answer versus more complex imaging applications. And we at Lumen Dynamics have invited Dr. Claire Brown to present on this topic in order to help researchers answer this simple yet complicated question. So, like I said, we welcome all our researchers, microscope companies, as well as Excite partners. And just a bit of housekeeping, in order to ensure that you're able to hear the presentation clearly, we have muted all your phone lines. Of course, we welcome your questions, and the way you can submit this is by typing them out at any time during the webinar by clicking on the chat box on your screen and direct it to George Q, who is the organizer of our meeting. At the end of the webinar, we have reserved 15 minutes in order for Claire to answer your questions. Also, we will be recording this webinar, and we will notify you by email when you can access this recording on our website. I strongly encourage any comments or feedback about the webinar or suggestions for any future webinars that you might have. We would like to know what real researchers out there want, and Lumen Dynamics will do our best to deliver. Now, without further ado, I would like to introduce our speaker, Dr. Claire Brown. Next slide, please. Claire Brown is an assistant professor at McGill University and the director of the McGill Life Sciences Complex Imaging Facility, where she has worked for the last 5 years. She oversees the operation and use of 13 state-of-the-art microscopy platforms, where approximately 150 users from 60 laboratories across Montreal conduct their microscopy research. She has expertise in many areas, including live cell imaging, turf microscopy, FLIM, and multiphoton microscopy. In her research, Claire has also been involved in the development and application of correlation microscopy for the measurement of protein concentrations, protein dynamics, and protein-protein interactions from fluorescence microscopy images. She has also developed and run more than 30 workshops and courses in collaboration with corporations in the field of light microscopy, including the inaugural Montreal Light Microscopy course in 2010, as well as the upcoming Cytometry and Microscopy Symposium, which is in November, and that's organized by the Canadian Cytometry Association. We very much appreciate the time that she has taken to prepare and present this webinar with her very clearly busy schedule. Claire, thank you very much, and I now pass it on to you to talk about which light source is best for your microscopy application: lamp, LED, or laser. Claire?
Okay. Thank you very much for that kind introduction, and I welcome everybody here this morning. Let's see. Okay. So basically, when I started thinking about this presentation, I thought we really need to have the light sources in context. So I'm going to spend a little bit of time just on some background of what does the microscope need to do in order to answer your research questions, a little bit of background on fluorescence and the fluorescence microscope. And then I'll go through an overview of all the different types of light sources, and then finish up with a comparison of the different light sources based on certain applications. So basically, the microscope needs to do three things. It needs to magnify the specimen so that you can see it. You need to have contrast, though, so you can't just magnify it. You also have to have a contrast between the specimen and the background, for instance. And most biological specimens do not have a lot of contrast. They're actually quite transparent. So there's a number of different ways that you can develop contrast. And then you have to be able to resolve the features you need to see. You may just need to be able to resolve how many cells do I have, or you may have a higher resolution application where you might need to say how many vesicles are within my cell, or what's the substructure of the mitochondria within my cell. So in order to have a reasonable microscope image, you need to actually do all three of these things. And I'll just illustrate this quickly with a quite visual example here. So this is an example of an image where you have lots of magnification and lots of contrast, but you don't have very much resolution. So it's fairly difficult to see the features within the sample. This is an example where you have resolution and magnification, but you don't have very much contrast. So even though the features are well defined, you can't see them against the background. The third example here is where you have high contrast and resolution, but you have low magnification, so the objects are just too small to visualize. So really you need all three of these, magnification, contrast, and resolution, in order to get a high-resolution image that you can perform your research and answer your cellular biology or small organism type questions. I'm not going to say too much about magnification. It really, kind of ironically, in microscopy is not a factor that comes into play a lot. Magnification is important, but it doesn't really determine resolution and contrast so much as other aspects. So basically, you need to have a lens that's going to make the features large enough for you to see. So for contrast, there's a number of different ways to contrast. We have visible dyes. So this example here just shows an H&E staining of some different tissues, and you get different colors. The nuclei are kind of purple, and proteins show up as red. You can use phase contrast imaging, where you get dark and light areas of the sample depending on their thickness and their viscosity. You can use differential interference contrast, or DIC microscopy, where you get nice outlines, good sensitivity to edges, but a bit of a different look to them than you have in a phase contrast. And there's also a dark field, where you can use oblique illumination of the sample, and this is really nice when people are looking at small organisms, and you can actually see some sub-resolution features, because in this technique you're only looking at the diffracted light from the sample. So you may see small flagella or hair-like structures on your sample that would not be visible with the other bright field techniques shown here. So the main problem with these technologies is that they lack specificity. So they're very good if you want to look and see, the shape of your cell, the morphology of your cell, how fast is your cell moving, how many cells do you have. But if you want to know something about your protein of interest or what's going on with your probe in the nucleus, then these techniques don't provide you that information. So in order to do that, you need to move to fluorescence. And the main reason that fluorescence microscopy has thrived so much is because of the specificity. So you can use antibodies, which are going to label very specific molecules within your tissue or your sample. This is an example of the peripheral nervous system of a Drosophila embryo. And you can see in green is microtubules, red are the cells that are dividing, and DAPI is just showing the nuclei of all the cells within the Drosophila. So you can get a lot of information, and now you're looking at very specific proteins or very specific regions of the cell with the fluorescence. If you want to move more into living tissues, then you move to fluorescent proteins and molecular probes. So this is an example shown here with a fluorescent protein called Paxillin GFC, which goes into focal adhesion complexes. You can see some small spots here in the cells. And this is a live cell nuclear probe, here shown in blue, and then we're using MitoTracker red. You can see the nice mitochondrial staining here. So this is a 3-color live cell experiment that's made possible with these different fluorescent probes. So basically, if you can have your sample and you can find the labels that you need in order to label the proteins you're interested in, then the next step is how do you image that at the microscope. And I just want to give a little bit of background of the light path in the microscope, because it will be important when we talk about the comparison of the different light sources. So sort of a general schematic here showing the light microscope in an inverted architecture is you have your light source, which typically gives off white light. And then you have an excitation filter, which chooses the color of excitation light. So here, this is a green filter, choosing green light from the light source. That then reflects off a dichroic mirror. This is a mirror that reflects certain wavelengths and passes other wavelengths. So this mirror in this example is reflecting the green light up onto your sample. Your sample has a red fluorophore that's going to absorb this green light and give off red fluorescence. The red fluorescence is going to be collected by the objective lens. It goes through this dichroic mirror now, and then you usually have an emission filter. And the main purpose of this filter is to get rid of any kind of reflections or scattered light that might be in the system. So some of the white light from the light source or the green light from the excitation might get through to the detector. So the emission filter is also sometimes called a barrier filter. It selects the red light, but it also blocks any stray light that might get into the detector. And then you have a detector, which is a camera or photomultiplier tube, depending on your system. So this is kind of more what it looks like in the real world. So this is an example of an upright microscope where you have a light source back here, which is a Mercury HBO lamp. And then the light comes through a number of different filters and other components, which I'll talk about briefly in a minute. And then in this case, the fluorescence cube is sitting right here. And you see the green light going down onto the sample, the red light coming up. And then you can have some other optics here with some beam splitters and so on. So in this case, you can choose whether the light goes to the eyepiece and you observe the sample by eye, or the light can go to the camera and you collect your image on the computer. This figure is really showing the same thing, but I'm just looking at an inverted architecture. I know people are probably working on one or the other type of microscope. So this is the same idea. We have the HBO lamp. You have the white light coming from the lamp. In this case, we're looking at a green fluorescent dye, so we have blue light being selected to go up to the sample. That light's going to excite our green fluorescent probe, and then the green light is going to come down through the dichroic mirror. And this one is actually showing a number of different ports, so you often have many different places you can send the light on your microscope. So this would be a side port where you could put a camera, the front port where you could put a camera. This is the path up to the eyepieces to observe the sample, and then you can also put a camera here. If you have what's called a trinocular, you can put a camera here as well. So lots of options as far as how to collect it and image the data. So if we look at a little bit more detail at the epi-illuminator, this is really the part of the microscope that sends the light to the sample. You have the light source again, here showing a Mercury HBO lamp. You have some collector lenses, which basically just focus and shape the light. You may have a heat filter, which I'll talk a bit more about after. If the lamp has a lot of infrared light component in it, you may need a heat filter to minimize that. There's often neutral density filters, and what these do is just they neutralize the light from across the whole spectrum. So the heat filter will specifically filter out infrared light, whereas the neutral density filters will just reduce the light completely. They're almost like having a pair of sunglasses where you just put them on to reduce the total light that's getting to the eye. And then at the end of this epi-illumination turret here, you have the turret containing the filter cubes. And typically the filter cubes are mounted with 6 or 8 or 10 cubes, and they can be rotated in and out of place when choosing the different dyes that you want to image. So just a couple more words about the fluorescent cube. It's shown here. Again, there's 3 components. You have light coming from the light source into an excitation filter. You have your dichroic mirror or your beam splitter, and then you have your emission filter or your barrier filter. And as I said before, that fits in this turret here. So basically what happens if we go back to the schematic that I showed you earlier is we can just put the filter cube in place here. So now this exciter filter here is the one shown on the cube, and that selects our excitation light. The dichroic mirror is at a 45-degree angle in the middle of the cube to reflect the green light and pass the red light, and then you see the emission filter, the barrier filter in this part of the cube. So I'll move now into the, this shows the spectra of the lamp. So this is the intensity on the Y scale here and the wavelength on the X scale. And you can see the color coding here going from violet to blue to red. And you'll see that the lamp has a very high component in the infrared, a lot of light out in the infrared, and then a fair bit of light in the visible. And this light is also temperature sensitive, so that if you change the temperature going across the tungsten filament, it actually changes the spectra of the lamp. So you'll see at 3400 K you have a lot more infrared light, but your visible light has tripled or quadrupled. So typically when you're working with bright field imaging, you work with the temperature at 3200 K, and that gives you the most white type of light for your sample. So for each light source, I'm going to show you some of the pros and cons. So this is a white light source. It offers light across the visible spectrum. It's very inexpensive, and the bulbs last a very long time. As I said, it's mostly for bright field illumination, but you can use it for fluorescence excitation. And I'll mention it again a bit later, but it's very good for live cell microscopy because you don't have any light in the ultraviolet, and the intensity of the light is relatively low. And you need to be aware that the color changes with temperature, especially if you're doing any quantitative imaging. You want to make sure you use it at the same temperature each time. So the second light source is probably the most common one on the, I don't know about common on the market now, but it's certainly the most common in use. It's also a white light source, and it uses an electrode that has two electrodes sealed under high pressure, and there's mercury contained in this in the electrode itself. And it's actually the burning of this mercury that causes the arcing and the light to be produced by the lamp. So the main advantage of the mercury lamp, or the HBO lamp it's sometimes called, is that it's 10 to 100 times brighter than the tungsten halogen lamp. So there's a lot of light. The light is also focused, so the light comes from a small arc that forms between the two electrodes. So it's a very dense light source. The photons are in one small location. You can get different powers, so you can go from 50 to 200 watts. And this shows the spectra here of the lamp, which you'll see is much more complicated than the spectra for the tungsten lamp. And basically, again, we're going from ultraviolet up to infrared. And the main difference here you'll see is that there's these large peaks at different wavelengths across the spectra. And a lot of the traditional standard fluorophores were developed just for these peaks because this was really the main light source that was used at the time. So a lot of the dyes that are 546 and 405 were designed to make use of these intense peaks. They're readily available, so you'll find them in most labs. Now there's some cons to these systems as well. So the bulbs last for about 200 hours, and then they have to be changed. When you do change the bulb, you have to do an alignment of the bulb within the lamp housing to get uniform illumination. And the bulbs are hazardous waste because they contain mercury, so they have to be disposed of properly. The lamp intensity also decays over time. As the mercury burns, the space between the two electrodes increases, and that causes changes in both the intensity and the stability of the light coming from the arc. The intensity across the spectrum has these nice bright peaks, which you can look at as a pro if you want to use dyes that match up with those peaks. But you can also look at it as a con because using the same exposure time for different dyes will result in a very different density of light being delivered to the sample. So you just need to be careful with that. The bulbs can also flicker, especially as the bulb ages. Again, the spacing between the two electrodes widens, so you get flickering or slight intensity variations on a fairly fast time scale, which can affect quantitative imaging. They also generate a lot of heat, so you need to make sure that heat gets dissipated, and if you're going to be doing long-term experiments, you may need to put the lamp on for a little while to equilibrate because it will heat up the microscope as well. So there is a fairly long warm-up time. It should be warmed up at least for an hour before starting experiments. And the excitation wavelengths can't be controlled independently. So you can't just say, I want to turn on the blue light now and excite my green dye, and then I want to turn on my green light and excite my red dye. You have to have some sort of external control of filter wheels or filter cube turrets in order to choose the different wavelengths for multicolor application. So the Xenon lamp has a very similar architecture to the mercury lamp. You can see the two bulbs down here. The Xenon lamp has a similar electrode architecture here in the bulb, and now it's a bulb under xenon gas. And you'll see that really changes the spectra quite significantly. You have a nice even spectra in the visible range, and then you have this huge component out in the infrared. So again, it's a white light source, so you can excite most dyes with it. Relatively even spectrum across the visible. They're 75 to 150 watts. And again, it's a focus light because the light is formed at this small position between the electrodes where you have the arc forming. And these are also fairly readily available. For the cons, the bulb life has a bulb life of 1,000 hours, so it's a little bit better than the Mercury, but they still have to be changed fairly often. The bulbs do have to be aligned, although some of the companies come with you can buy the bulb in the housing, which just gets substituted right into the lamp housing. They are still hazardous waste, so they have to be dealt with properly. And the lamp intensity still decays over time. So it's the same sort of reasons, that the electrodes are actually burning, and as the distance between them changes, you have the intensity of the arc decays. You have weaker intensity in the UV, so you could see that as a pro or a con in the sense that if you're doing imaging with UV excitable dyes, you may need more power from the Mercury lamp. But if you're doing live cell imaging, this may be a pro. They generate a lot of heat because you see this huge amount of light in the infrared area. So normally you should have a heat filter in place if you're going to use one of these on your microscope. And the power is not so high in the visible range in comparison to the Mercury. And the same as the Mercury lamp, the excitation wavelengths are not controlled independently. You have to use some sort of filter wheel or filter cubes, changing in and out in order to select specific wavelengths for excitation. So the next lamp is the metal halide lamp. And if we start over here on the spectra, you'll see it has a very similar spectra to the Mercury lamp. The Mercury lamp is shown in red, the HBO, and the metal halide is shown in yellow. And what you'll see is the peaks are pretty similar. Some of them are lower, some of them are about the same height. But the main advantage of the metal halide you'll see here is that the space between the peaks, the powers are much higher, especially here in the region where you would have all of your standard green dyes. You have about a 3-fold increase in the brightness of the lamp in that region. So it is a white light source. It's brighter in between these Mercury peaks. There's no bulb alignment, so these are architectured a little bit, they're built a little bit differently. The bulb actually goes into this sort of a housing which is actually external from the microscope. And then this liquid light guide is used to connect the bulb to the microscope, and the light goes through the liquid light guide to the system. So the bulbs are much more stable over time, so you don't have as much decay of intensity. The bulbs last about 2,000 hours, so that's 10 times longer than the HBO bulbs. And because you're coupling to the microscope with this liquid light guide, the field is very uniform. So in the Mercury lamp, even after your alignment, you always have the center of the field of view is a bit brighter, and then it decays as you go out to the edges. And you can deal with this by using control samples and correcting for shading. But with the liquid light guide on the metal halide, you typically don't need to do this because the field is very uniform. Less heat compared with the HBO and the Xenon, and the heat is isolated in this box away from the microscope. The other thing is the intensity can be controlled directly, so you can actually say I want 1% or 5% or 10% of my lamp power. With the Mercury lamps, this is usually done manually with a control pad if there is one, or it's done using neutral density filters. And some of these systems have shutters and long-term feedback so that the shuttering can actually be done within the lamp housing instead of having to have a shutter on the microscope. And this can be good if you're worried about any vibrations on the microscope system itself. And there's long-term feedback, so this makes sure that if you're looking at 5% power, that if there's any fluctuations in the light source, it's still running at the same total power density. So I just had those two examples I mentioned earlier. Here you can see if you were using excitation, a green excitation for, say, a rhodamine probe or something, the difference between these two lamps would be very minimal. Whereas if you were looking at blue excitation for a green dye, you see this threefold increase in intensity with the metal halide. So some of the cons, they are more expensive. So if you're buying a mercury lamp, it's going to be more affordable than the metal halide lamp. The bulbs cost more, but they also last longer. So in the long run, the costs may even out. Sometimes the liquid light guides have to be replaced over time. And what we do on our systems is we keep the shutter inside the lamp housing closed when we're not using it for the microscope, so that the light is not going through the liquid light guide unless we're actually imaging on the microscope, and that helps prolong the life of the liquid light guides. So you do require a shutter to block the light. It doesn't switch on and off automatically. And you can't control the excitation wavelengths independently. Similar to the HBO and the Xenon, you'd have to have a filter wheel or filter cubes in order to specifically select different colors for excitation. So the next light source is LEDs, or light-emitting diodes. And LEDs are basically individual optics that can have the variable colors. So instead of having one bulb that's a white light source, you actually have an LED for each color within the spectrum. And sometimes these are coupled to the microscope directly, and sometimes they're coupled by a liquid light guide. So it depends on the technology. So there's quite a few pros of the LEDs. They can be built right into a microscope stand because they're quite small. They have discrete color peaks, so this is showing the emission peaks for various LEDs across the visible spectrum from the ultraviolet to the infrared. Excitation filters are not always needed, so you can see here for this violet light or this blue light, their bands are quite narrow and be very similar to the type of band you would get using an excitation filter. But you'll see here in the yellow and the green area that these bands are quite broad, and you may need to put in some kind of a bandpass filter to select just the peak of these bands. You can do direct fast switching, so you can turn each LED on and off by itself, so you don't need to have shutter in front of the light source. There's many wavelengths available. This is just showing the visible area, but there are LEDs below 400 nanometers, and there's also some up above 650, I believe. There's no intensity decay over time. The light's either on or off, and if the LED fails, it will just fail, and there'll be no light. The bulbs last for about 10,000 hours, and it's my understanding that 10,000 hours is the time that it's on. So they're actually only on when you're exposing, because they can be switched on and off so quickly. So this is an extremely long lifetime. There's little heat generated from them. You can control the intensity of each LED independently, so you could have one at 10%, one at 100%. If you're running the mercury lamp and you're using ND filters, it gets a little more complicated if you want to attenuate different colors in different ways. There's no warm-up or cool-down time, so basically when they're turned on, they're at full power. And some systems can be programmed for automated imaging, so you could set it up to do 3 or 4 color time-lapse and walk away from the system. So there are some cons. If you look here, this is the first spectra I showed you, and these spectra are normalized, so you see that they're all going up to 100%. So that's 100% for that LED. The next spectra actually shows the LEDs with their powers, with their actual powers, rather than being normalized. So one thing you'll notice is that some of the LEDs further down in the blue have a lot more power than some of the yellow or green LEDs. So this is something to be kept in mind too, that if you do see a plot like the first one, to just take care to ask the right questions to the company and find out what the powers are for the different lines. So it's not a white light source. You have to combine a number of different LEDs to get the colors that you need. And as I said, low powers at some of these wavelengths in the green and the yellow. The green to yellow LEDs have this broad emission. They're fairly expensive up front, so again, compared to a mercury lamp, you're looking at about 3 times the cost. It depends on the architecture. The different companies are selling these kinds of systems, and some of them are limited by the number of wavelengths. You can only pick 3 or 4. So if you're in a lab that's using the same 3 or 4 dyes all the time, it may be fine. But if you're in a core facility or something like that and you want to have a lot of flexibility, sometimes that can be a bit of a limiting factor. So I'm not going to say as much about gas lasers, but I want to be complete in the discussion of light sources. So I'm going to give a little bit of information about lasers, but these typically are more suited for laser scanning microscopes than wide field microscopes. But I think the general points should follow. So the pros of gas lasers is that you get single wavelengths for excitation. So if you have a 488 nanometer laser, it's 488 nanometers, and there's no bandwidth to that or anything. It's exactly 488 nanometers. With the gas lasers, you can often get multiple lines, so you can buy one laser and be able to excite in the blue, the green, and the yellow, for instance. Most have lots of power, so you can do applications that require a lot of power, so we'll talk a little bit about applying this for photoactivation or photobleaching techniques. And it's a concentrated focused light source, so the power density is very high because the light is focused into a very small spot. The cons is that some of them are very expensive, depends on the type of laser, but you know, when you start having to buy multiple lasers, it can add up pretty quickly. They can generate heat. They have limited lifetimes, so they do decay over time, and then they tend to just fail and then have to be replaced. And they do have intensity fluctuations, so they need to be warmed up for at least an hour before use, and then you can have both short-term and long-term intensity fluctuations. The field of lasers has moved more into diode lasers now, and the reason for this, some of the pros, they're very compact. You don't have to cool them. They have long lifetimes, so from what I understand, they'll either go in the first year or they might go for 5 or 6 years. Higher powers are now available, so when these were first developed, they didn't have a lot of power, but now I think there's 300 milliwatt 405 lasers now in the diodes. They tend to be very expensive, and similar to the LEDs, you need to have one for each color, so it can add up quite quickly. There's no warm-up period, again, when they're turned on, they're on, and there's minimal intensity fluctuations. They are sensitive to electrostatic charges, so you have to be careful you don't destroy it by giving the system a shock. And as I said, there's only one color per diode, so if you want to do multicolor imaging, you need to buy multiple diode lasers. So the last class of lasers I'm just going to mention is infrared and white light lasers. So the pros of these systems is that they're tunable to multiple wavelengths. So you can tune the laser cavity basically to change the color of the light by moving different elements, different mirrors, and so on. In the early days of these systems, it was quite difficult to tune them, but now most of this is computerized. So you would just go into the computer and say, I want to tune the laser to 488 nanometers, and it should do that for you. They have extremely high powers, so that's certainly not a limiting factor for anything. The infrared lasers are used for multiphoton microscopy, and they can penetrate deep into tissues. Up to, I think, close to 1 millimeter has been demonstrated now. The cons are that they require a pump laser, so you have to have a second laser to produce lasing in the IR and the white light lasers. They're extremely expensive, so they're upwards of $150,000. They're technically challenging to use and maintain, so you normally need somebody around who's going to be around on a regular basis and keep them tuned and aligned. And they generate a lot of heat, so you may have to do modifications to your workspace, your microscope room, in order to deal with the heat. And they typically require large optical tables because they're coupled into the microscope directly. They're not coupled by fibers. So the last little bit, just on the light sources in particular, is a little bit of a power comparison. So I just wanted to bring up a few terms. This table shows the power for the different light sources in a number of different units. So the first one is called radiant flux, which is basically the total power output of the system. And you can see the tungsten halogen lamp has a lot of power output, and it's actually a higher power output than the mercury lamp. But the radiant flux is all of the light, so a lot of this light is in the infrared region where it's not as usable as it would be if it was in the visible spectra. The luminous flux is then the power in the visible spectra. So you see here that the tungsten and the mercury even out a bit more. And then I think the most important, two of the most important things to keep in mind with any of these light sources is the spectral irradiance. So this is where they take the power and then look at it per unit area and then per nanometer. So it's how much power density do you have at the different wavelengths. So you see for the tungsten halogen, this one goes way down to less than 1 milliwatt per meter squared nanometer. And this is because there's so much of that power spread out into the infrared. And the other reason is that the light source is very big. So the tungsten halogen has a tungsten filament within the cavity of the lamp, which is very large. So there's a lot of light, but it's not focused. So this is really a light density when we're saying how much power do we have per meter squared. So the combination of the wide spectra and a lot of light in the infrared and the large size of the source make the spectral irradiance of the tungsten halogen quite low. In turn, with the mercury lamp, you have this very small source size. So it's an order of magnitude smaller than what you have in the tungsten halogen, and it's just that arc between the two electrodes. So you see here the spectral irradiance is much, much higher. The other point here is that the LEDs, they don't have a lot of radiant flux or luminous flux because they're only giving a very small bandwidth of light. But the spectral irradiance is actually fairly comparable to the Xenon lamp because that light is focused into a small area. So for that color of light, you actually have a fairly decent excitation power. So the main point I want to make with this table, and actually I can mention I have the websites at the bottom of all the slides if I've used material from the web, so feel free to go there and get further information. But the main point for this slide is to, when you're taking into account, think of the fluorophores you want to use and look at the power for the given wavelengths that you need, not just the overall power for the light source. And how concentrated is the light? So is it a focused light source or is it a larger light source like the tungsten halogen? And the best thing to do is measure the power at the sample. So there are devices you can purchase where you can just put a power meter at the sample and look in your microscope with your lenses and your optics, how much power am I actually getting to the sample. So I'm just going to go now to some application-specific comparisons of the different technologies. And I don't think I'll have time to go into a lot of detail into each of these tables, but you'll have the information, and we could discuss a bit in question time as well. So what I'm going to do is just tell you the reason I think which is the best choice for each technology, and then I'll maybe just mention a couple of things about the other light sources. So the main issue with quantitative imaging is you want to collect data in a uniform way across space, time, and between samples. So the LEDs are a great choice for this because they have uniform illumination across the field of view in space. The lamp is stable over time, so you have stability between time points or between samples. And you have precise control of the intensity, so you can regulate how much light gets to the sample. So that would be, I think, the best choice for quantitative imaging. Now that being said, if you're on a limited budget and you can't afford to buy an LED light source, then some of the other light sources, the mercury, xenon, metal halide, would be fine. The metal halide has the advantage of the uniform illumination. If you have a mercury or a xenon lamp and you don't have the metal halide, what you can do is just put on a standard fluorescent plastic slide and take an image of your field. and see the curvature of your field, and then use that image to correct, to flatten the background, or shading correction type algorithms can be used. So it's not that you can't do quantitative imaging on the other technologies, it's just that the LED would be the preferred. If we go to live cell imaging, the main issue there is you want to minimize all light exposure, and in particular you want to minimize heat from infrared light, and you want to minimize ultraviolet light as much as you can. So again, the LEDs are a nice source, because if you choose to do excitation with blue or green LED, there is no ultraviolet component. Whereas on the Mercury or the metal halide lamp, you have quite a bit of light in the ultraviolet. In the Xenon, you have a lot of light in the infrared. The other advantage is that in the live cell, you want to use lower powers. So the LEDs give you a lot of flexibility, and the fact that they're lower power is actually seen as an advantage. However, again, if you have an HBO light source and you want to do live cell imaging, it doesn't mean you can't do it. It just means that you have to add filters like ultraviolet filters and ND filters in order to minimize the light exposure, and then use a shutter to control the exposure time. For photo bleaching and photo activation, the main issue is that you need high power, and the power needs to be focused and concentrated. So lasers are really the best choice for that, because you can focus the laser into a small spot, and you can use acousto-optic devices to turn the laser on and off and do pattern photo bleaching or pattern photo activation. The LEDs right now don't really have enough power to do a lot of photo bleaching experiments, but they're continually working on the technology and getting higher and higher power sources. With the other light sources, the lamp sources, you can do some photo bleaching or photo activation by using the field diaphragm and minimizing that so that you can bleach, say, the center of the field of view if you center the object of interest in the middle of your field of view. So I'm going to finish up on multicolor applications, and I just wanted to give a couple schematics just showing how you can do multicolor. So the most sensitive way to do multicolor is by using three filter cubes, where you would move the different filter cubes in place to take your blue image, your green image, and your red image. And this is fairly slow because it's limited by the movement of the filter cubes, but it's the most sensitive way because you're selecting out just the colors of light that you're interested in. So it's good sensitivity, good resolution, low crosstalk between dyes because you're only imaging one dye at a time, so you're not going to have your green dye going into your red channel because it's not being excited at the time that you're collecting your green image. But as I said, it's slow. And you would basically take 3 monochrome pictures with your blue cube, your green cube, and your red cube, and then the computer pseudo-colors those images and overlays them into your 3-color fluorescence image. If you want to do multicolor imaging and you want to go fast, then there's 2 ways you can do this. You can use a triple filter cube. So this is a cube that will have an excitation filter that will pass 3 different wavelengths, a dichroic mirror that will reflect 3 different wavelengths and pass 3 different wavelengths, and an emission filter that will pass 3 different wavelengths. These cubes are fairly standardly available, so you could have the blue, the green, and the red fluorescence. And basically you're going to lower your sensitivity, and the best example is if this mirror has to reflect a green light source in order to excite your red dye, then some of your green fluorescence coming back to the detector is also going to be reflected by this mirror off this way and won't be detected. So you will lose sensitivity, but you gain speed. So you also, if you do this 1 fluorophore at a time, so if you take the LED example, if you turn on your blue LED and then your green LED and then your red LED, then you would image the 3 dyes sequentially, and you would avoid crosstalk again between the dyes. And it's still going to be faster than the last method because you don't have to move the filter cube in and out. So if you're using a standard lamp like an HBO or so on, you usually have to use filter wheels in order to choose the different colors of light on the excitation and the emission, and that usually takes on the time scale of seconds. You can get faster filter wheels that will go about 10 to 100 milliseconds, but not much faster than that. For the LED, all you have to do is turn the LEDs on and off. You don't have to put the different filters in place. You could use a triple excitation filter and then just turn the LEDs on sequentially. You can do something similar with lasers. And I'll mention just briefly a monochromator, which basically allows you to move a mirror to choose the different colors. So the fastest way to do multicolor is to do it simultaneously. So you would turn on all three colors of light at the same time and image them simultaneously. So this is very fast. Again, you have the lower sensitivity because of the multi-coated dichroics. You do get crosstalk between the dyes, so you have to do your proper controls. And your resolution will depend on how you do the detection. So if you use 3 cameras, so if you could put in some optics here to split off the blue, the green, and the red light to 3 cameras, that would give you high resolution, but it'd be quite expensive. There are single chip color cameras you can use, but they have lower resolution and lower sensitivity. And there's also a 3-chip camera where it splits the light within the camera, and that would give you high resolution, but it's expensive. So for the multicolor, basically it depends if you want high sensitivity or speed. If you just want to do high sensitivity, there's really no advantage to going to the other detectors because the main advantage is speed. So with a Mercury lamp, you could do fine 3-color imaging with 3 fluorescent cubes. If you do want to go fast, then it's the LED and the lasers that will allow you to do that fast switching. And if you want to do it simultaneously, you just have to make sure you do good controls. So just to summarize, I have a table just comparing the different applications with little plus signs. If anybody agrees or disagrees with any of my plus signs, I'd love to talk because this was just something I put together. So for quantitative imaging, the LED and lasers are the best because, well, the LED is really the best because it has the stability and power over time and the uniform field illumination. For live cell, the LEDs are good because you don't have the UV or IR components. And the tungsten halogen, actually, I didn't mention, is also good because it doesn't have that UV component. You can use the other light sources, you just need to make sure you put in the proper filters. For photobleaching and photoactivation, really lasers are the best choice, and LEDs aren't quite ready for that yet. For multicolor applications, you can use pretty much all of the technologies, but if you want to go fast, then it would be the LED or the lasers that would be preferred. And that's what I have, so thank you for your attention, and I'd be happy to take any questions.
Thank you for that, Claire. If we have any questions, please make sure you type them in the chat box on your screen. And I'm going to start with a couple questions that have come in, Claire. The first one is regarding photo bleaching of samples. Which one is worse, white light or lasers?
Oh, that's a tough question. It really depends on the dye, and on your scanning principles of your laser and so on. So in general, I would say the laser is worse because it's a focused light, and the photobleaching is mostly due to molecules accepting another photon when they're in the excited state or in the triplet state. So lasers are probably worse just because the light is focused in that small spot. But then you don't have the ultraviolet component and so on if you're using it. So it's fairly complex, but off the top I'd say the lasers because of their density.
Okay. And then we have another one. What is the limitation of LEDs in respect to fish when about 5 to 6 colors are needed?
I think the main thing would be if your light source is able to deliver that many colors. So I know some of the systems on the market, you can only have 4 LEDs at a time, and then if you want other ones, you can swap them in and out, but you can't use more than 4 at a time. There's other companies that are putting all of the LEDs into a light source similar to the metal halide light source, and then all of the lights coming out of fiber. And so you're not limited. You can then have 7 or 8, and they're calling those white light sources. So I think you'd want to make sure you've got the right technology. And if you had the right technology and were able to make the filters, it should be possible.
Okay. And on that note about the light guide delivery, there's a question of which one is better for imaging direct or light guide coupled?
I think the main advantage of the light guide coupled is the uniform field. So I would say the light guide coupled is better because you have the uniform field, you bring the heat away from the microscope, and if it's the shutter controlled, then you also get rid of any vibration that would be associated with the microscope.
Okay. And then for super resolution, this viewer has said that they've heard you can do super resolution with white light or with LEDs. Is that something you can comment on?
I haven't heard of that. The general issue with super resolution is the power. So if there's enough power from the LEDs to photo bleach or photo convert the dyes, then it would be possible. So it's just a matter of whether you have enough power. So I'm not aware of those applications, but I'm sure that's the direction people are moving.
Okay. Oh, and is there any benefit of liquid light guide over quartz fiber?
That I don't know. I don't know much about quartz fiber.
There's another one that says, hope you'll have the presentation downloadable from some website. Yes, we will have the presentation downloadable, and we'll let you know by email because we are not sure when it will be ready. But when it is, you can probably go to our website to be able to access it. And I think at this point, we will end the session unless there are any more questions. On behalf of Lumen Dynamics and Excite, I would like to thank all of the attendees, and a really big thank you to Claire for enlightening us on this subject, and thank you, Claire, for taking out the time from your crazy schedule.
You're welcome.
And if there's any feedback, please do let us know. If there's any other webinar we can do for you, please let us know. Our details are on the screen. If you have any more questions for Dr. Claire Brown, please address them to her directly or to myself, Kavita Aswani. And again, thank you for your time, and wherever you are in the world, have a good day.